talk about Bayesian model for sparse regression when there is high-dimensional data. So this is a joint work with Sylvia Richardson and actually Jeff Rosenthal. So what is the problem that we have? So in general, people would like to understand the relationship between the high-dimensional data set, right? And they would like to understand the relationship between like predictors, big set of predictors, and big set of like outcomes. Now, what is the approaches like a statistician are apply to reduce the dimensionality of this problem? Well, I just list a couple of them. So you might try like to reduce basically the dimensionality of the outcomes by principal components, and you do a regression on the principal component. You can do like the independence component analysis, which is similar to the PCA. You can also think in terms of clustering, rather than like try to find like the association between the X matrix and the Y. You try first like to cluster the Y matrix into subgroups, and do the association into like each single group that you have identified. Or you might also want like to understand which is the relationship between the Ys, the outcomes. Maybe you want like to do like a graphical models on that. So you discover basically the partial correlation that exists. Now this is like for dimension reduction you can apply, but people might want like to understand the relationship between the predictor and the outcomes without using this set of like a dimension reduction technique. They want like actually to understand precisely which is the association between each single predictor and the outcome. So people use like a regression model, right? I will be like define like precisely later on what type of regression model. For a large set of responses and predictors, and this is like a technique that allows like to identify precisely what is like the association. However, the problem is when like the dimension of Y grows is very large. Technique that like try like to infer the relationship between the predictor and the outcomes is not feasible, right? So we have like to invent something different. So this is like the technique that like it's like on the pocket of like a statistician in order like to do like association and dimension reduction. What we have done start like actually from a biological problem. So I don't want to get into like details about the biological problem, but like a biologist, I think like to discover association between some genetic quantity and other genetic quantity. There is not like a idea of causation, but the relationship between one quantity and others. So for instance, like biologists are interested like on identify the association between DNA levels or DNA like characteristics of each individual, and another like a quantity that is like a biologist are interested in, which is like the expression. So they want like to identify the relationship between your DNA, and if DNA like infer or like can cause like a level of genetics level. They might also be interested like in other type of relationship. For instance, we just change like the title, MQTL. They might be interested like in association between your DNA of the participant of the study, and their metabolic level. Now, there is not just one gene or just one metabolite. The number of genes is around 20,000. So what like a biologist is interested in is like to identify relationship between the DNA and like the 20,000 genes, or like the DNA and like the 1,000 of metabolites. So this is like the type of, and we want like to find the pattern of this relationship. So this is like the type of, and also there is like a different type of example, like for instance, like they want like to identify the relationship between copy number variation and gene expression and so forth. So this is the quantity that biology is working with. Now, we are not like working with all these examples. We have specifically one example, which is EQTL. So the EQTL, what it is, so biologists want to identify the relationship between your DNA, right? You have like a DNA level, which has been measured, and the production of the gene expression. They want to identify if there is a relationship between the activity of your genes and like the characteristic of your DNA. Now, as you can think, this is a big dimensional problem because imagine that like the predictors are your DNA. The DNA is like around 1 billion base pairs, right? Even like if we are not analyze all of them, we just like find like the one that are different between individuals, they are like high dimensional. So the space of the predictors is huge. And what they want to do is like to associate this high dimensional predictor space to another high dimensional response space, which is the gene expression. Okay, and this is called EQTL. So you see that now the dimension is huge predictor space, huge outcome space. However, the number of individuals in this study is relatively small. So here we are like in the usual setup of small n, large p, but also if you think, also large q, where q is a number of like genes. So everything is big, but like the number of observation. The biologists want to uncover like the multivariate association without using any like clustering or dimension reduction technique, because they want to go like back to the laboratory and say this part of your DNA predict this particular gene. They don't want to say they predict like basically a principal component. They want to measure it in the laboratory. Another thing that they are interested in is the following. I try like to be clear. So what I told so far is like the biologists are interested in association between particular part of your DNA and the genes. However, part of DNA play a key role. So some part of DNA control not just one gene, not 10 genes, but can predict like, thousands of genes. So this is called hotspot. So we are part of our DNA, which actually regulate at the same time many genes together. And this is primarily important for biology, to identify this part of your DNA that regulate the activity of many genes together. Okay, so this is the intro. So how we try to solve this problem? Well, I'll try like to skip like the details, but now we have a huge set of predictors, which is X. We have a huge set of outcomes, which is Y. And what we are interested in, actually the dimension of X is n times P, n subject P predictors. And the dimension of Y is n times Q, where Q is the number of responses. So what we are interested in is basically to understand the relationship between each single predictor with each single outcome. So what we are interested in reality is like to understand the structure between the association that we can be encoded by a matrix which is Q times P, with Q is the number of the outcomes, P is the number of predictors. And if we know this matrix, P times Q, we can associate every single pair between SNP and genes. In order to understand this, or like to make inference, we create like a Bayesian hierarchical models. I will like detail later on. In order like to search for the large number of predictors, we use like a method that we developed like a couple of years ago, evolutionary stochastic search. And actually, probably like the most interesting part of my talk today is the following. It's true that like the biologists add, like in the analysis, thousands and thousands of genes to be associated with like the level of the DNA. But not all like basically the Q genes. But not all the Q genes are in reality associated with like the DNA. Only a couple of them, only like a subset of them. So when we do like a model that try to find the association between the predictor and the outcome, sometimes we spend in the algorithm too much time try like to find the association that doesn't exist. So the idea was the following: why don't doing like the algorithm we learn which are like the genes that are not associated with like the DNA. So basically on the fly, where like we run the algorithm on the fly, we learn which are like the non-important association. And basically we spent more time basically to try to find out the association on those genes that actually are important. So basically this is like a learning algorithm with the MCMC. So you see that here, using this trick, we don't do like clustering. We don't like remove part of the genes of the outcomes. We use all of them. It's doing the algorithm that we learn which are important. So I mean like there is an association between DNA and genes, and those that are not important. Okay? A little bit of notation. So the notation is the following. So we have our big matrix Y, right? Dimension is n times Q. N is number of subjects. Q is number of the outcomes. Q is the order of 1000, right? The predictor is like the matrix X on the same subject, nth time P, where P is the number of predictors. P is much larger than N, and Q is larger than N. Now, for a particular like outcome, K, right, for a particular K, which is like a particular genes K, we can try like to find the association between our predictor and like the particular outcome. How we do it? This is a linear model, right? So we just, like I said, that like we predict the value of the gene K by a linear model using all the predictors, and the error term is like a normal distribution. Now, how we perform like the selection of what are the important part of your DNA that predict the gene K? Well, we extend like the model space, the state space, and we add basically a variable gamma K, which is like a binary variable that says, the X column of X is important to predict the YK or not. So it's like a binary. So we have like a vector gamma K, which is a vector of binary values, which get like the value equal to 1 if the regression coefficient is different from 0. Otherwise, it's equal to 0. The idea on all like this like variable selection method is like to make inference about this vector here. But now this vector here is just for a particular K. Now you think that K goes from 1 to big Q. So actually, if you want, you have to perform Q of this like variable selection experiment. So at the end, as I told you before, the inference, the primary goal of like our method is like to make inference about this vector gamma here. With actually for all the K's, all the Q-like responses is like a matrix, big gamma. This is just like, like a way how we specify like the value of the beta, the regression coefficient, which is like G prior, which is a technical element. Now, the hierarchical model, the Bayesian hierarchical model has to be completed by like the final part. And the final part is the following. What is the probability, which is like gamma kj, means like what is the probability that there is an association between the jth predictor to the kth outcome, okay? What is this probability? Well, the probability, we have like in the Bayesian setup, we have like to say like is a distribution probability, is a Bernoulli distribution, which is conditioned on this value here, which is like the prior probability. So the prior probability can be like encoded into like a Bernoulli distribution with probability omega kj. Now, before I was telling you that like we are interested in this matrix, but the probability to observe a 1 is actually encoded like in this matrix here, omega. And this is like the first result that I want to present. So now we have like to specify which is our prior belief about this like a probability. So the probability of the Bernoulli distribution. The Bernoulli distribution is the one that is 0 or 1, meaning the association between like the DNA and the transcript. Well, how we specify this? Well, there are 3 different ways to specify this. So the first specification that we can use is like on the top, number 1, says the following, says like the prior probability that there is an association between the jth predictor, the jth element of your DNA, and the kth transcript, the kth genes, right, is simply omega k. So you see that there is no influence now by like the jth predictor. So they are independent for each like outcome. The second one says that the other one, the way around, said like the probability of association is basically provided just like by like the probability by like the DNA location. So here basically said that like this joint probability just influenced by like the transcript. Here said just influenced by your DNA at the position J. So what we have done actually, we extended these 2 models, say that actually the probability kj can be factorized into 2 parts. One which is like the probability to observe a 1 for each like transcript, multiplied by a factor rho j. So rho j says the following, said we have for each transcript we have some probability of observing 1s and 0. But actually when we look at a particular combination of kj, this probability is inflated or deflated by this factor rho j. And this factor rho j basically said that like we can have like some probability, but this probability can be inflated because like there is a larger probability if you want to see like the association between the K transcript and J like DNA level. Okay, so the inference that we have to make is like about this quantity and this quantity a posteriori. This is like the model that I just described. The MC strategy for this type of model, well, it's like quite complicated. The quantity that we are interested in is like this vector of association, gamma, 0 and 1. We are also interested like in evaluating like this probability a posteriori, which is the probability of association. And we are also like interested like in find like the value of the betas, which is the regression coefficient. The scanning strategy, which is the second part of like the innovation that we develop, is the following. Now we have a Q, very large number of transcripts. And for each of them, of outcome, we can perform like a variable selection, as I described. We get like basically a posteriori the value of this gamma, the value of this omega. But if you want like to perform like a MCMC, this will take quite a lot of time. Why? Because you have to scan, go through all the possible Q responses. So you basically do like an algorithm that like scan from the first transcript until the Q transcript. One strategy that you can use, for instance, is like to instead of using like scanning all of them, you can go like in a subset of them, right, with equal probability. So for instance, if you want to save a fourth of your, actually 3/4 of your time, you just select at random with equal probability 25% of the transcript. On those, you perform the variable selection, you estimate the quantity that I told you before. But as I told you before, is it like a valuable strategy, this one? Well, basically, this is not like take into account that sometimes you don't have important association between like the transcript and the DNA level. So basically, you just scan all of them irrespectively of knowing that there is no like an association or learning that there is no association. So we adapted a adaptive strategy that I'm going to illustrate. The adaptive strategy is the following. During the MCMC, we learn some weights that are indicated with WK. And this weight depends on a coefficient B, and the coefficient B is like the batch index. So during the MCMC, we're going to perform the variable selection, right, on those transcripts that shows like that have a higher level of probability to be selected. Now, what is like, this trick? So basically, instead of sampling uniform at random, we sample with this probability without replacement. So we basically spent more time doing like the exploration of the relationship that I described before on those transcripts that shows higher probability or higher weights. How we learn this weight? Well, the algorithm works as follows. Every 50 iteration, the bell rings. We stop our like MCMC, and we look like at the past path of what we have like analyzed so far, or what is the output so far in history of our chain. We construct basically this weight based on some past value that we recorded of our MCMC. Now, the definition of batch, this is a little bit different respect to what people are using like in adaptive MCMC. There are two conditions that has to be fulfilled, technical condition. The first one said, okay, what is the definition of batch? So the past history. The definition of past history is the following. It should be like not only like the value that we recorded like on the previous 50 iteration, but should like, like the history should go to infinity as the value of B goes to infinity. So we have like basically to keep all the past history as soon as like we start like the algorithm like proceed. The second technical condition is the following, that what is like, how much information the batch should share during like the MCMC. Well, this second condition said that when B goes to infinity, the overlap between two consecutive batches should be equal to 1. So basically, at infinitum, they have to share basically all the same history. So this is two technical conditions of batch. People that work on adaptive MCMC, they don't like use this condition. They just look at the previous 50, let's say, iteration. And they base like their adaptive proposal using just like the closest history of the chain. Here we have these two like a technical condition. Finally, what we are measuring actually. So we are measuring something. We don't have like a particular target here. What we measure, like in the past, is this quantity, which is RKB. So K is like for the KF transcript or the KF outcome. R is some quantity that is informative about if this KF transcript is important or not. So if you have to spend more time and do variable selection on this particular transcript. For instance, but it's not like limited to, you can measure the number of variables that are the model dimension that we explored in the past. Example, if the model dimension is equal to 0, means like this transcript is not important. So it will provide less weight. So basically, we learn that it's not important. We don't spend much more time. And actually, where are we going to spend more time? Well, these are the weights are larger, right? And when it comes, well, when, for instance, like the model size, instead of being like small, is very large. So for those transcripts where the model size is large, basically this is like informative about the weights, and we spend more time on this in this transcript. Okay, I have just 2 minutes. I hope like to say there are some more technical conditions here, but like I'm skipping. Just like to give you like an idea of how the algorithm works, the second part. So this is a graph of the weights, right? So how evolve over time. For up to some batch index, we do not like actually evaluate this weight. Or actually, we do the following. We let the algorithm learning, but we select this weight with equal probability. So the up to this batch index, they are like the weights are all the time the same. So well, basically, we sample with equal probability. Soon after, we have quite a lot of history. And we use this history to inform about like the probability. You see that actually in black is the one that actually we simulate, is a simulated example. We know that actually there is some signal, right? So you see that actually the algorithm learn that actually this particular outcome, K, is quite important. They give a lot of weights, right? While like the pale like gray is the one that there is no association. So the algorithm should like learn that it's not important. Okay, this is like a fine because the majority are like on the lower part. Well, few of them have like weights that are greater than like the uniform one. This is like for instance, what happens like for 2 particular trajectory. The algorithm can also like learn at the beginning, can struggle. This is like for simulated with association. Can struggle at the beginning to give like the correct weight, but after a while, it increases. So actually, if we let them run like more than 2,000 iterations, probably all the dark one goes up and all like the pale gray goes down. Okay, I finish. A little bit of like a summary, conclusion. So the problem that we face today is like the association between a big y-dimensional matrix and an x-dimensional matrix. What we have done is basically we use like standard techniques of variable selection, right, to reconstruct the association between these 2 big dimensional matrices. The problem is Q might be very large. We don't want like to do any clustering algorithm beforehand because we don't know which are the important ones. So we have an algorithm that actually discriminates between the important one and non-important one. What is important and non-important? Well, it depends on your definition of R, right? It can change. You might be interested not on like the amount, the dimension. You can be interested like on the coefficient of determination and so forth. Okay? Thank you.
Hi, that was great. Thank you.
Thank you.
So I wanted to ask you about one of the particular things you mentioned during when you were describing the adaptive scheme.
Yes.
How expensive is it in terms of both time and memory to record the entire history instead of a small? It sounds difficult, is it not?
Well, in reality, true what you said, but we have a trick. And the trick is like in reality, we can discard. So you see that like the time history grows, grows, and grows. But actually, we can even like start like to discard the original part. But we can like discard like the original path of our MCMC. But it depends on which rate we can discard. It should be like bounded. So we can discard like the square root of B original history of the chain. Cannot be linear, otherwise basically, but we can in reality like discard part of the history. And this saves quite a lot of memory. sublinear, but it saves quite a lot of memory. As well as we start to record this like trajectory from the beginning without burn-in. So you may imagine that at the beginning, the algorithm is very stupid. So it's very good actually to start discarding as soon as like we move on the original like record.
Okay, thank you.
Okay.
Yeah.
So yeah, that's very interesting. So in the adaptive kind of adjustment of the selection weights,
Yes.
is it motivated by any sort of theoretical reason that those weights are near optimal for particular types of models, or
No,
it's
there is no optimality, unfortunately.
okay.
No, there is not
It's like a
optimality.
heuristic sort of.
It's yes, actually, your question is why, like in adaptive, in, people that use adaptation, they target, the optimal acceptance rate. There is an
optimal.
Exactly,
Yeah, so
but you see here we do not have any optimality, so we don't target anything, right? That is like a disadvantage. The advantage is actually we can construct this function R, whatever we want. So for instance, at the moment, what we are doing is we have a huge set of variables that or like characteristics of the MCMC that we are recording. Model size, coefficient of determination. We model like the entropy. We have a bunch. And actually, we construct basically a combination of them that can be informative about like R. And then R is informative about the weights. But you are right, there is no optimality in this. Thank you.